I just wanted to start by saying that I know we don't have a patient speaker here tonight. I think it's probably not a secret to most of you that I'm also a patient. And when I first moved to Charleston to start graduate school in 2018, the first provider I had here was Susan. If you don't know Susan, she's a physical therapist here, and she had told me about these meetings. And so I came here during my first semester of graduate school in the fall. It was right when I was starting in the lab. Didn't know I was going to study EDS yet, but this meant a lot to me because this was the first place I ever met someone else with EDS. Really, Susan was the first person I met with it after I was diagnosed and in person, and this was the first place I met anyone else. So this is really cool to be here as an expert and speak tonight. So I included this graphic because I found it the other day and I think it's really awesome. The reference is at the bottom. If you want to find it, it should be free to access. This is one of the best summary infographics I've seen for EDS before. There are a few things that we could probably pick out and say they're not perfectly accurate, like maybe the information on prevalence, but I wanted to include this here because this is a really great resource that I just found. I don't really need to explain what EDS is to most of you. It's a group of connective tissue disorders. Depending on who you ask, they'll say there's 13 or 14 types. In the 2017 classification, there were 13 types recognized. Since then, another rare type of EDS was identified. So that's why I go with 14, even though the classification didn't recognize it at the time. Main features: joint hypermobility, skin hyperextensibility, and tissue fragility. It's generally viewed as an extracellular matrix or collagen disorder. You can see in this graphic the process of collagen being processed, and I actually show this a little bit later as well. For all the subtypes of EDS, except for HEDS, there are known genetic markers, meaning you can get genetic testing and be diagnosed with one of the rare subtypes. However, that's not the case for HEDS. We're obviously hoping to change that, but right now, a genetic test is not going to tell you that you have it. So hypermobile EDS. Prevalence of EDS in general used to be estimated at 1 in 5,000 people. We definitely know that's not true. A paper was published a few years ago that showed 1 in 500. That doesn't account for people who are undiagnosed, but that 1 in 500 number is similar to the prevalence of things that everyone has heard of, like MS, cardiomyopathy, Parkinson's disease, things that have research going on all over the world, and most of you have probably heard of them, but that's not the case for EDS. We know that there's a spectrum, so patients may present pretty mildly with sort of joint hypermobility, maybe an occasional dislocation, and then you have patients who have a really severe phenotype, lots of comorbidities, lots of hospitalizations, medical procedures, they may need mobility aids, and some patients may even end up bedbound at periods of time. So one of the big issues is that we don't have that genetic test, right? So there are these delays in diagnosis. It's estimated to be about 14 years. We see that women with HEDS tend to present more severely or are more likely to be diagnosed, and the phenotype tends to worsen around puberty. So you hear a lot of patients saying that, maybe when they were in middle school or high school, they suddenly had these symptoms that they were dealing with, but they weren't diagnosed yet. They were probably diagnosed 10 or more years later. So because of all of the different manifestations, it really requires care from a diverse team of medical experts. And this is just an overview of what some of those things can look like. This is not everything, of course. We have the joint issues, the skin issues that you're probably familiar with, things like hernias, pelvic organ prolapse, cervical instability, Chiari malformation, and then the comorbidities, so gastroparesis, POTS, mast cell activation syndrome. There are more, but this is maybe what an EDS patient might actually look like. There was a worldwide survey that was done a few years ago. This was published, and this is where they found that on average it took 14 years to be diagnosed. So 14 years from when your symptoms started until you received an accurate diagnosis. And 56% of patients said that they were probably misdiagnosed in that process. 86% of patients said that that delay in diagnosis resulted in worse outcomes. So maybe they had symptoms that weren't being treated right away, maybe they had a surgery or a medication that they didn't actually need that caused harm. And again, 70% of those misdiagnoses resulted in inappropriate treatment. So maybe being misdiagnosed with an autoimmune disease or something like fibromyalgia where you were given a medication that actually wasn't for the issue that you had. 35% of patients experienced that healthcare professionals refused to see them. So they had a diagnosis of EDS, or a suspected EDS diagnosis. They wanted to see genetics. Genetics said, well, we don't see HEDS patients. You hear stories like that in a lot of different areas. The main reason patients felt that they were being denied care is because they were viewed as being too complex of a patient. And here are a few quotes from some other publications that I'm sure will resonate with a lot of you. One of the things that patients emphasize is a failure of empathy in provider-patient relationships, where they felt like their healthcare providers were not empathizing with them or understanding what they were going through. They also struggled with clinicians who didn't have knowledge on EDS. Another thing that people saw was an issue with awareness and education. I'm sure everyone in this room has probably seen a doctor who had to Google what EDS was while they were sitting there. And they also found that EDS really needs to be a focus of research. So these are what patients are saying they want to see, and it really supports everything that we're trying to do. So the big question is, where are these issues coming from, and how do we fix this? How do we improve diagnosis so people are diagnosed earlier? How can we advance patient care and develop treatments? And you really see this cycle of there's a gap in clinical care, and from that, to fix it, we need interest from physicians and scientists who want to study this. And those people will develop research questions, who will then apply for funding to make those discoveries. And those discoveries go back to educate the current physicians or the next generation of physicians and scientists and inform clinical care. So by going through that circle, we eventually make progress. And I think our lab and the group here at MUSC is trying to tackle almost every one of these circles. So I'll tell you a bit about that. But I did want to talk a little bit about some of the past hurdles. So if you go into PubMed and you do a search for just Ehlers-Danlos syndrome, and you look at the years that papers have been published, when you look prior to 2003, there were 2,000 papers ever published, or at least on the internet. I don't know if people were publishing it before then. And there's only 33 that are focused on hypermobile EDS before 2003. When you look at NIH funding, from 1985 to 2003, there are 163 projects that mention Ehlers-Danlos syndrome somewhere in their grant application. So they might say, we're studying aortic aneurysm, and it occurs in EDS, and so that comes up in the search. When you focus just on hypermobile EDS, there are 0. And when you look in the last 20 years of publications, we see 2,800 papers. And when limiting it just to HEDS, we see 339. So that trend is improving. I mean, you can see it just from the picture here. It's getting better, but we're not quite where we need to be. And with NIH funding, in the last 20 years, we've seen 2 projects that are actually focused directly on EDS. These are focused on vascular EDS, but still EDS. And when you look at hypermobile, there aren't any. But I did just get a score on my fellowship grant, which is focused on hypermobile EDS. It was a very good score that should get funded. So there will be a grant on HEDS. And there will be more, looking at you back there. Hopefully, so a postdoc grant is great, but a faculty level grant is a really big deal. And so Chip is hopefully going to get one of those soon. And then we can give this talk another time and we can say there are 2. And there's been a lot of progress sort of in media attention, right? So some of you have probably seen some of these celebrities being diagnosed with EDS sharing their story. Jamila Jamil, Halsey was a big one, Sia, and some local stories from the Post and Courier, the MUSC Catalyst, and a CNN article that came out on Christmas Eve. That was actually one of the top 100 stories in CNN this year. So clearly the public is listening, engaged, and interested, but research in medicine really needs to catch up. So one of the big issues is we don't know much about the underlying biology of HEDS, and even the types of EDS where we know the genetic cause, research has sort of stopped there. There isn't much work going on into developing treatments or understanding the biological consequences. There are very few labs and physicians studying EDS, despite there being so many researchers that study connective tissue and collagen. They don't really narrow in their focus on EDS or HEDS, and we're hoping to try to change that. And then the other big gap is that many patients struggle to find knowledgeable care. You might have to travel really far, you might have to spend a lot of money, you might have to pay out of pocket. Patients even move. I mean, I've heard of people moving across the country to live near knowledgeable physicians. So there's still a lot we have to do. And when you think about our knowledge of EDS, I like to say that we're just starting to go up that slope. We know more than we've ever known before, but there is so much left to learn, and I think we're about to move in that huge growth direction, hopefully. So our lab uses this model, the cycle of discovery, to sort of talk about how we view research and how we view making translational discoveries and going back and making an impact with patients. It really starts with patients who have something, who share their experiences with physicians who collaborate with us. We can collect genetic and biological samples, we can generate animal models to study disease mechanisms, and then the ultimate goal, of course, is to find diagnostic tools and therapies that go back and help patients. But there's a lot more that goes on in some of these areas. When it comes to interacting with physicians, there's a lot of different things that we're doing, and I'll talk more about some of those in a little. And when we talk about animal models and disease mechanisms, that is where the bulk of the time is spent. So getting genetic samples, doing sequencing is actually fairly fast and easy. Understanding how different gene mutations cause changes in connective tissue is much, much harder. And then figuring out where you can intervene for therapy is one of the hardest things we have to do. And I mention this because I know a lot of people have heard about some genetics discoveries from our lab and have been wondering, where's the paper? When are we going to know what the gene is? Things like that. We are in the process of getting that published, but one of the reasons it took so long is to validate that biologically. And I'm going to talk a little bit about why that's important and why it's needed. But first, I'm going to give you a little background biology. So genetics, study of genes, genetic variation, hereditary, how things are passed down from generation to generation. Your DNA is the genetic material in your cells that tell your body how to make things. You can think of your DNA as an instruction manual. So if the instructions are wrong, no matter what you're trying to make, it's not going to be perfect. And nucleotides are the building blocks of DNA. So the A, T, G, and C, that it's actually, right here in the image, that make up your DNA. So when you talk about a mutation, you might have a T where an A is supposed to be. And that DNA is replicated and transcribed and then translated into protein. And that's where our big focus is. When you have a mutation, how does that affect the protein's function and cause disease? And so just a little bit more background. Alleles are different forms of the same gene, so you get one from each parent. I think most people have heard about this when we talk about inheritance and how things are passed down. A mutation is a change. And this is where I like to emphasize mutation does not always equal disease. The genotype is the alleles that you have present, and a phenotype is the trait of that person. So if you have a genotype that causes an eye color and your eyes are blue, blue eyes is the phenotype. So these are the other subtypes of EDS. HEDS is in there. It shows that the genetic basis is unknown. You can see the genetic basis of other types and then what protein is involved. And also the inheritance pattern. So when you see something that's autosomal dominant, that's when we're talking about HEDS. Typically, one, or in some cases, both parents are affected, and it's passed down to the child. Both parents do not have to be affected, though, for a kid to have HEDS. And then with recessive, you have parents that are carriers. And so there's a chance that one kid will get a copy from each parent and then have the disease. So some of the very rare types of EDS are recessive, but the more common ones and HEDS are believed to be autosomal dominant. And then genetic testing. So we get a lot of questions from patients about genetic testing that they may have had and if they can look for mutations that our lab has identified. And it really depends on what you've had done. So one of the tests that you can get done is a microarray. If anyone's ever done 23andMe or Ancestry, that's an example of a microarray. If you look at the sentence here as your whole genome, and each word is a gene, a microarray looks at specific letters. So these are specific SNPs, locations, where it's looking at the genotype. It's not getting the whole picture. With whole gene sequencing or an exon array, if anyone's ever had a connective tissue panel for EDS, this is the type of testing they use. This might be looking at individual genes, but it's not giving you the whole picture of your genome. And then whole exome sequencing is looking at the parts of your genome that code for proteins. So that would be every single sentence is sequenced, but you're not sequencing the spaces in between because not really sure that they're important for this, right? They're not making proteins. If you have whole genome sequencing, that is absolutely everything. It's sequencing the entire genome. It's an overwhelming amount of information, but it can be potentially useful, especially in research environments. So when patients ask us, should I have a connective tissue panel if it's assumed that I have HEDS? I always say yes, and that's because you don't know if you have another type of EDS. The symptoms can overlap. It's always good if your insurance will pay for it, if you have access to genetic testing, get it done, rule out anything else. If it comes back normal and you meet EDS diagnostic criteria, you still have HEDS. It does not remove your diagnosis. That being said, it only looks for known genetic mutations. So that means anything we're working on in research isn't going to show up there. If you use direct-to-consumer testing, 23andMe or Ancestry, and then you try to look at your raw data, it is not going to be diagnostic for EDS. There's a lot of situations where patients try to do this. I know there's like a frequent miscall in Ancestry where it tells everyone they have vascular EDS. It can actually be really anxiety-inducing, and it's also not diagnostic quality sequencing information. So along with that, it can miss something entirely. So again, like I showed you back here, it's only looking at these specific letters. And so it could actually miss another type of EDS as well. And when it comes to whole exome or whole genome sequencing, you get a lot of info. It can be a lot to digest if you don't have the help of a geneticist or genetic counselor or someone who can counsel you on this information. It's usually not covered by insurance, but one of the benefits is if you have this information and tomorrow a paper is published on a genetic cause of HEDS, you can go look at it because you already have all of your data. So there are pros and cons. As I said, genetic mutations are common. They're not all bad. Evolution is driven by mutations and changes like that. Even at birth, you will have anywhere from 100 to 300 different mutations that neither one of your parents had. This occurs frequently. Throughout your DNA, you'll have anywhere from 4 to 5 million SNPs. So if that tells you anything, that's why it's been hard to isolate a gene for HEDS. That leads me to talking about everyone's favorite thing, variants of uncertain significance, or VUS. So a lot of patients with suspected EDS will have a connective tissue panel ordered. And they will find that they have a variant of uncertain significance in maybe collagen 5 or ZNF or some other gene involved in a type of EDS. And the doctor usually says, we don't know what this means, there's not enough information, and sends you away. A lot of people come to us then and ask us, what does this mean? And we pretty much give you the same answer. A variant of unknown significance could be completely benign or actually be causing disease, but without validating what that genetic consequence has on the biology, there's really no way to know. And unfortunately, that's a big disconnect between clinical genetics and research, is that geneticists will see a VUS and don't have a lab or aren't affiliated with anyone who can do any follow-up testing, so patients are left with this VUS. Keeping in mind that these are only known genes for connective tissue disorders, so you could be hung up on a VUS when there's something else in your genome that we don't even know is causative yet. So as I mentioned, genetics is important, but biology is where the real important stuff happens. So your DNA is eventually transcribed, translated, becomes protein. What are the biological consequences of a genetic mutation? That's where we start looking at what does this protein interact with? How does that cause changes in connective tissue? It's not just simply collagen's messed up. And I know there's the question sometimes, every once in a while, can we just take collagen supplements? I wish it was that easy. But there's a lot more going on, and if your body's not making connective tissue components correctly, your collagen might not function properly, even if there's not a mutation directly in a collagen gene. So when we talk about mechanisms and pathways and what's going on in a cell, sometimes you see diagrams like this, right? One thing comes here, something inhibits this, goes inside, comes out. Simple. It actually looks like this. And this is why the biology is the hard part. And when you talk about connective tissue, you might see this like cute little drawing. Here's collagen and elastin, but it actually looks like this. These are actually really cool. If anyone wants to look at awesome science art, this artist makes really cool images. So the big focus of what we do in the lab comes from understanding this stuff. So I'm going to give you an example of something that we've been working on and how we're going through genetics, biology, and mechanisms. and then also looking at some comorbidities. So our research on HEDS really started with one large family. You can see those with an EDS diagnosis are shown in black in this pedigree, and circles are females, squares are males. The gray is probable HEDS. Most of you probably can imagine what that means, but in this case, it was someone who was younger, so hypermobile, but weren't totally sure if they definitely had HEDS. They had some signs of comorbidities, and then older individuals, right? So they're stiffer, they're not going to meet the Beighton scoring criteria anymore, but they have a history that's consistent with they probably have HEDS. And so we took the approach of doing whole exome sequencing, and I don't think you can see this. And we did that on this person right here and this person right here. So the benefit of that is that they are second cousins. So when you're looking for something that's causative, they share about 3% of their DNA. So you're gonna be able to narrow down that massive amount of millions of SNPs to a much smaller number. And so that left us with 48,000 shared variants instead of 5 million. And the next step is to use different prediction platforms and databases out there to try and narrow down what could be causative. We got down to 6 rare and potentially damaging variants. So since we had the entire family, we sequenced the whole family for all 6 of these, and we only found one that was present in everyone who was coded as black and gray in this diagram. So when we did that, there was one that tracked, and this was in a gene previously unknown to be involved in connective tissue biology, codes for an enzyme, and unfortunately I can't tell you exactly what that is at this time, but hopefully soon. Hopefully you'll see a paper coming out of this and everyone can learn about it then. But I always like to remind people, even if you knew what it was right now, it would not change anything about your care or anything like that. So how do we show that this mutation in a gene, not a collagen gene, something completely unknown to being connective tissue and related, how do we show that's causing HEDS? Well, the first thing we looked at is it expressed in tissues that matter, right? If it was like only in the kidney, we'd probably be like, maybe this isn't right. So we wanted to see that it was broadly expressed in different tissues, things that are connective tissue. And when we looked into some databases, we saw tendon and skeletal muscle and arteries and skin, things that were relevant to HEDS. And so the next step was to do some simple biology in cells. So all we did is we took the normal copy of this enzyme and the mutated one, and we put it into cells, and we said, make the protein, see what happens. When we did that, we saw that with the mutation, right here, all of the enzyme that needs to be out in this extracellular matrix and interact with collagen and other things to function gets stuck inside. And a normal person, it's coming out and being secreted. We still don't know what it's doing out there, but we at least saw it's getting stuck inside. And so we did that right here. This is called a Western blot. Our interns have run many of them this summer. And you can see that here's the wild type inside the cell and the mutant's getting stuck in there. And outside the cell, the wild type is coming out. That's what all these black marks are. And the mutant is not. So that was showing us that at least inside cells, this mutation seems to be causing a problem, right? So we're kind of taking it from that VUS space of we don't know if this is important to it looks like it's causing a problem. It might be pathogenic. The next thing for us was to make a mouse model. This is one of the best ways to validate a genetic discovery. In this case, you take a mouse that's completely genetically normal, like all other mice, and you put that one single mutation in it. So if it has problems, you know that it's coming from what you put in there. And this allows us to say, yes, we're correct, no, we're wrong. But if we're correct, then we can use that mouse to study the progression. We can take tissues from that mouse. We can test therapies on that mouse. It becomes a really awesome tool in our research. So we went ahead and did that, and we got mice from the CRISPR-Cas9, which if anyone's ever heard of it, it actually won the Nobel Prize last year, which is pretty cool. It's not our technology. Really cool, smart people figured that out. And so how do you know if a mouse has EDS? And we were like, oh, they just like go through the EDS criteria. But they walk on 4 legs, right? So let's cross that one off. Apparently mice are super hypermobile. You can take their foot and bend it all the way back to touch their shin. That's how they like crawl into little spaces and all the stuff that mice do. Their knees already hyperextend in a normal mouse. So we had to be a bit more creative about how to approach if something was wrong with this mouse. One of the first things I noticed, so the way you handle a laboratory mouse is you pick them up by the tail. That's like the appropriate way to move them. And I went to pick up a couple mice, and they were able to turn all the way around and bite me. Or try to bite me. I didn't get bit all the time. But I mean, that had never happened before. So I was like, oh my gosh, like their spines, they must be hypermobile. So we got a bit more creative, and we decided to focus on 2 tissues where we did sort of a histological readout and a functional readout. So we took the Achilles tendon, and we did that because in the original family that I showed you that we identified the mutation in, there was a history of Achilles tendon rupture. It's also a pretty large tendon in the mouse, so it was like an easy thing for us to take out and focus on. And we worked with some people in bioengineering who took that tendon and they put it between a clamp, kind of shown like this machine right here. And they slowly stretch it over time, and you can measure the mechanical properties of that tissue and see how elastic it is. And then we did transmission electron microscopy, which is a way to look at a really, really, really high magnification what collagen fibrils look like. And this is a picture of that. So in our normal mice, you can see all of these collagen fibrils. There is normally variability, so some are going to be bigger than others. But when we take a look at the mutant, we see a lot more of these small fibrils everywhere. And we actually do have data quantifying all of this. I didn't want to shove too much scientific stuff at you today. But we saw the mice had smaller fibrils. And we also saw that their tendons were more elastic when we stretched them. And when we looked at the heart, we saw that the mice had mitral valve prolapse. We could see this on staining, and we could see it on echocardiography. And so we had 2 different tissues where we validated, hey, these mice are different. And if you want to add on that they tried to bite me, that's a third thing. We also saw that you could tie the tail in a knot in mutant mice. So it was another thing. It was like, hey, their tail is more elastic. So this was really great confirmation for us that what we were seeing in patients, we took this, we isolated it, and we actually looked at the biology and said, hey, biologically, these mice have a connective tissue problem. But we're still sitting here wondering, the gene we found is not collagen, but it affects collagen. How are we getting here? And so that's when we're starting to look at a mechanism. How does protein X play a role in collagen assembly or synthesis or processing? Are there other roles for this protein in connective tissue beyond just collagen? I mean, I showed you that big mess of stuff. It could be in a million different things. And so I just wanted to show you a little schematic of sort of how collagen is made and synthesized and then secreted outside of the cell. So a lot of things go on. I'm also really proud of this picture, so I really wanted to use it. So eventually, after it's translated and it's hydroxylated and glycosylated, you get these triple helixes of collagen. And that goes and gets secreted from the cell and comes out, and the propeptide gets cleaved, and now you have this collagen that's almost ready to be put into those fibril circles that we saw. But first, this enzyme called lysyl oxidase has to come in and get it ready to cross-link into these big fibrils. And then you get those circles that we saw. And so since we saw those smaller collagen fibrils, that led us to the idea that maybe there's an issue in cross-linking. These fibrils aren't getting put together properly. So some of the work in our lab has been focused on understanding a role for lysyl oxidase, and I'll mention that a little bit later. That's something that Taylor, a PhD student in our lab, is working on. We also are doing some additional genetics. So I said that's one family, right? That doesn't explain everyone. We wanted to know how common is this, what else is going on. And so we did an initial study of 198 patients, unrelated individuals. We did whole exome sequencing. And we did a lookup for this gene and also the family of genes that this gene is part of. The genes have similar functions, similar structure, and we hypothesized that if you mess with any of them, you could get a similar issue. So when we did that, we saw that around 33% of these 198 patients had a mutation in this gene family. And so all of this, along with some more data, is actually what is in the publication that we've submitted. So to get those 198 patients, we developed a patient registry. This was for genetics, but also to look at some clinical things. We've had over 7,000 patients fill out the pre-study questionnaire, which is awesome. And we've consented almost 2,500, and we have almost 2,000 DNA samples right now banked in our lab. One of the perks of that is that we can do large-scale genetic studies, and I'll mention that briefly, but I did want to share some of the clinical demographics that we got out of this. So pretty in line with what we expected. We actually saw a large percentage of our patients were female. We also saw a large percentage had a family history. And this is the prevalence of some of the different phenotypes. So you can see in yellow, these are, those things that are part of the HEDS diagnostic criteria on that 2017 checklist. And then in the green are things that are shown to be associated with HEDS, but not necessarily considered in the diagnostic process. We saw a lot of things that necessarily weren't surprising, but things that I think were really interesting. Only 61% of patients have joint dislocations. So I think that kind of dispels that myth that you have to dislocate to have HEDS. Maybe you just haven't dislocated yet. And maybe having a diagnosis could prevent it. Chronic pain was super high. Anxiety and depression were really prevalent. And GI manifestations were present in 82% of patients, yet not considered at all in diagnosis of HEDS. When we break all of these things down between males and females, we see a higher prevalence of almost everything in females, a few things that were higher in males. And then we did a cluster analysis, and this is something that Taylor is also working on in collaboration with someone in statistics, to see if we saw any specific sort of separations of almost subtypes of presentation of HEDS. And there were 3 clusters that were able to be identified. And when you look at these, you can see the 3 clusters, and then you can also see this overlap of how they're all represented. What was really interesting is I found that there was really sort of a typical HEDS meeting that 2017 criteria cluster, and that was this yellow one, right? We see the highest prevalence of subluxations and dislocations, we see wound healing and stretchy skin, and we see chronic pain. But then when we look at maybe this cluster over here, we see this higher prevalence of, although it's not super high, we see a higher prevalence of tethered cord and Chiari and CCI. So almost more of a more neurological or neurosurgical cluster. This information is really interesting because as we do more genetics, we might be able to pair specific genetic information with specific phenotypes. So maybe there are gene variants that cause patients to have more of a mast cell phenotype, and that's why some patients maybe don't really have that because they have a different underlying genetic issue. So I'm going to tell you a little bit about some of the other ongoing research projects in our lab. When we started, this was one project with one family with one PhD student and Dr. Norris, and that was it. And it's now turned into many projects, multiple students, our summer interns, volunteers, undergrads, postdocs, medical students, pretty much everything you can think of. And so we have a lot more going on now than we did 5 years ago. So one of the really cool things that we're working on is a genome-wide association study. This is a large-scale genetic project that helps us to identify common genetic changes in the HEDS population. We are working with a GWAS expert, Nabila. She is actually in Paris, so I've unfortunately had to go to Paris to work on some of this. With Taylor, we had a terrible time. We are going back a little bit more in the fall, which is really exciting, and that's actually the lab group that we worked with right here. One of the coolest things is that we got to show them all of this data, which is a lot more biology than what they do, and teach a lot of them about EDS. And so it's been a really great collaboration, and we are excited to go back. This was done on 1,000 patients with HEDS. So this should reveal some really interesting genetic things and maybe things that we can correlate to specific presentations of HEDS. As I mentioned, protein interactions are a really important part of what we're doing, understanding that mechanism. How do we go from random gene to collagen problem? And we did something called a 2-hybrid screen. It basically identifies things that this protein can bind with. This has been a focus of Taylor's PhD work and also Leon, a postdoc in the lab. And that's kind of where that lysyl oxidase I mentioned came in. It was one of our hits, but it wasn't the only one. Like I said, it's a whole network of all different things interacting, and so we're investigating how those play a role. Another focus, which is a project that I'm really interested in, is mast cell activation syndrome. kind of understanding, are mast cells maybe driving the disease? Are they causing problems in connective tissue? Maybe are mast cells getting activated as a result of the connective tissue being weaker? Is it a combination of both? Not sure, hoping to piece that together. But here is a video of mast cells degranulating for you. And there they go. So that's actually what's happening inside you when you have hives and maybe can't breathe. If anyone has asthma, that's also happening inside you. Biomarkers are another focus of our lab. We're collaborating with the EDS Society as well as some people in proteomics here at MUSC to understand if there are things that we can detect in the plasma or the serum of EDS patients that are increased or decreased that could serve as a biomarker for diagnosing patients, right? Like if you don't even need to get genetic testing, or you have a small child that you suspect might develop EDS because you have it, you can go to the doctor, you can get blood work done and get an answer. That would be super informative. So that's one of the things we're doing. Another project that we're working on is looking at dermal biopsies. So as most of you know, Dr. Patel does a lot of surgeries on EDS patients here. Initially, we had planned to take punch biopsies in the dermatology clinic, but then we realized that if we go to the OR, there is skin that is discarded every time an incision is closed. So we're able to actually put together an IRB that's being worked on by Victoria in our lab right now and collaborating with people in dermatology, Alan and Tyler. And from that, we're hopefully going to be able to identify specific cellular, transcript, genetic changes that might be going on in tissue biopsies from patients. Another thing, also, Dr. Patel is looking at tethered cord. There was a paper published by Dr. Klinge about a year ago, I think it was, maybe longer, where they looked at the phylum of tethered cord patients with EDS. They saw immune cells, they saw a change in elasticity of the phylum. It's actually the opposite of everything else in EDS. The phylum is less elastic. So what's going on there? Why is it happening? We have no idea. So we want to get phylum from patients. The problem is always finding control phylums from people who don't have EDS to compare to, because you can't take healthy people's phylums out. So that's something that's in the works. I think we might have found a way to get some control samples, so we'll hopefully be able to do that soon. Understand, why does tethered cord develop? I would love to be able to study this in the mouse, but the human phylum is really small. The mouse one, I don't think we'd be able to find. And I don't know if it even goes all the way into the tail. I'm not sure how that works. So this is where having human subjects that are with EDS who are willing to be involved in research, they're already having tethered cord surgery, right? You're just saying, yeah, take my phylum, use it for research, is really useful and impactful. We're also working on a project with vagus nerve stimulation. This is in collaboration with Dr. Kautz and Dr. Baudron and Dr. Boger. The idea is to use an at-home vagus nerve stimulation device that patients will be trained to use and record just initial outcomes of, what do you see a change in? Is your pain different? Is your POTS different? Is your GI stuff different? And then we can use that data, pair it with doing vagus nerve stimulation on our mouse model, and get very specific outcomes of how this could maybe be a potential treatment option for managing some symptoms of HEDS. Orthopedic outcomes is another project. Dr. Eichinger is an orthopedic surgeon here and brought up the question of, why do HEDS patients seem to recover differently? What might be going on after surgery or in the healing process that leads to poor outcomes? What can we do to improve those? A lot of patients with EDS need surgery. We know that surgery doesn't always work the way we want it to, so it's not always a solution. So having better treatments, better solutions, what can we do right now without having a direct therapy to have better outcomes? We're also looking at some of the GI complications of EDS. A medical student in our lab, Rebecca, is doing a chart review to look at what EDS patients at MUSC have dealt with in terms of GI issues. What procedures have they had? Have they had feeding tubes? What testing has actually been done? Is there maybe a gap in testing? Are we not doing motility testing when we should be, trying to identify anywhere that we can improve and also see what we can learn from these patients? Molly and Dr. Wilkerson are looking at some things with mental health. I showed you that anxiety and depression are super prevalent in HEDS. Why is that? Sleep issues, is that just because of pain, or is there something else going on here? And so we're going to work on trying to understand what's going on with some of that. And then we're putting together a new patient survey. So I showed you the prevalence of different things, right? How many patients have dislocations? How many have GI issues? But these questions, when we started our registry, were not very specific. And that was mostly because we thought we'd get like 30 patients. Never expected to have this large number of patients involved, and we never expected that we would be able to do anything with the clinical data. So we're now revising that to ask very specific questions to find out exactly what people's comorbidities are. Do they have other autoimmune conditions? Have they had surgeries? What has your experience been? How long did it take you to get diagnosed? How far do you have to travel for care? Because we have this large registry, we have the ability to ask a lot of questions that can inform funding, can help us advocate for funding from the NIH, can change clinical care, can inform research discoveries, all those things. And then one of the other really cool things we're working on is developing mouse models for every single type of EDS. I mentioned that even the types where we know the genetic cause, there has been little to nothing except for vascular EDS done on the biology or trying to treat it. So Dr. Norris had this idea of what if we could make every single mouse model and then make them available to other researchers for free to try and grow the number of people studying EDS. There are, I think it's 70,000 people studying connective tissue. None of them, maybe a handful, are focused on EDS. But they have the knowledge to do this stuff, and so if we can provide these models, maybe break down a barrier to cost or tools that people might need, we can get others involved in trying to understand some of this. And so that's been done with our transgenic animal core here at MUSC. We already have some of the models. One of our interns actually has been working on some stuff with it already this summer. And then one of the last things, wanted to talk about our mechanisms of funding and awareness, like what can we do outside of the lab, what can everyone else do to try and help? And so there are organizations like the Bobby Jones CSF, like the EDS Society, Dysautonomia International, other organizations for comorbidities that can be really helpful. There are foundations and donors who have made a lot of our research possible. But the big thing that can make a huge impact is an increase in NIH funding. Like I showed you, there's no grants focused on HEDS. I don't know if they're not being written or they're just not being funded. But this year we got language from MUSC, from our group and from administrative leaders on HEDS into the Committee on Appropriations for the House of Representatives to try and increase funding for HEDS. So this is actually the wording that we submitted, and if you look up the actual document, you can see all of this. But the goal is to really encourage the NIH to put money into establishing institutes that will improve clinical care and further research and just make HEDS a funding priority for the NIH. 1 in 500 is not a rare disease. There are people all over affected with this. We have these kinds of programs in place for things that are actually more rare than HEDS. And so this was a really big deal. This was really exciting, and we're continuing to work with people at MUSC who can help make a difference in this, because this isn't just NIH funding for our lab or me as a postdoc. This is funding where they can make a funding announcement and say this is a priority. And so researchers who need money for their lab can say, okay, well, we study connective tissue, let's submit a grant on HEDS to this funding announcement. So it's a really cool opportunity to grow what's going on in the research space. But even without addressing research, we know there are huge hurdles to care. Right, there's not one specialty. No one with HEDS sees one doctor. If you do, I need to know who that doctor is. You have to travel, right? Even living here and having some great doctors here, I've still traveled. When I lived in Pennsylvania, I traveled all the time for care. It's expensive. Travel's hard, right? If you're sick and you're I need to see a doctor because I'm so sick, I need someone who gets this. And then they're here, take a plane and then a 2-hour car ride and then stay at a hotel. Also, your parent or your significant other or someone has to take off work to go with you. It's hard. And if you are someone who's in a family of people who are affected, you might be taking the whole family. So there's really nowhere that you have all these experts in one place. And it's not just hard for patients, but it's hard for your physicians, because your neurosurgeon might do your tethered cord surgery, but when you're sitting in the hospital having severe GI issues afterwards, and there's no GI doctor who gets it at the hospital you're at, you're still stuck, even though you got that one issue fixed. And so having people in the same place allows physicians to collaborate on that care. So we really have been talking about how there needs to be this one-stop shop for EDS, and I've been talking about this with Dr. Patel and Dr. Norris and other people at MUSC for the last couple years about how this really needs to happen. So I think there's hope coming our way, and I say that because we're working towards trying to develop an institute here at MUSC. I'm hoping that it doesn't just happen here, but it happens in cities all over the country. The idea is to have a comprehensive care center that includes all different specialties for clinical care, but it also includes research, and it also focuses on education. Educating healthcare providers, nurses, doctors, physical therapists, educating medical students, and then having people like our intern program coming in and learning how to be the scientists kind of leading the future of EDS research. Right now, we are in the hiring process for a medical director, and ideally, once that's in place, we'll be able to grow into other specialties and have experts working together to understand how to take better care of their patients, but also to inform research. So having those physicians reach out to our lab and say, hey, we're seeing this in our patients, can you guys look into it? Or having those physicians say, hey, I think I want to take biopsies from my surgeries, do you guys want them? And having that constant translational research environment. And earlier today we were having a conversation and Dr. Norris said, every medical university says, researchers and clinicians, they work together. We focus on translational research. But it really doesn't happen like that very often. And so this is a really unique opportunity to be able to do that. And so with that, I just want to give a little shout out to everyone else in the lab. We have a pretty large lab right now. This is one of our recent photos. We have 5 interns in the lab who are all here right now as part of our EDS intern program. We also have an intern program alum sitting back there. And then we have a lot of collaborators at MUSC who have been big driving forces for a lot of this. Our collaborators in Paris and at MGH and Harvard. If you want to hear more about our research or stay up to date, we have like a news section on our lab website. If we have any publications, any events, anything happens, we put that information on there, thenorrislab.com. I think if you go to Norris Lab without the, they study like birds or something, so that's not the right one. Also on Twitter, there's like a bird person, Norris Lab. It's the Norris Lab on everything. They beat us to it. And I'm also Court Does Science on Instagram, Twitter. I tried to do a TikTok thing. I'm not as good at that one, but the other two. Yeah, so I want to let you guys ask any questions, and yeah, that's it.